Welcome to the Blueprint. Today, we are looking at how to extend the healthy lifespan of a mammalian cell, preventing it from freezing and turning gray in a state of cellular senescence. The target of this protocol is a specific protein called interleukin-11, or IL-11. It is a cytokine, a microscopic cell signaling molecule that naturally drives inflammation in the body. We'll systematically map how it moves, track its effects as tissues age, and deploy a specific method to block its signaling pathway. Neutralizing this single gene halts the runaway scarring of tissue fibrosis. It cuts off the supply lines that feed certain tumors, and in laboratory mice, blocking it has been shown to extend a healthy lifespan by up to 25%. Step 1 is understanding how IL-11 attaches to cells. To do that, we have to locate its primary docking station on the cell membrane, the IL-11R alpha receptor. It sits next to a co-receptor called GP130. In what is known as the classic signaling pathway, an IL-11 molecule drops down and locks into place on both receptors. This triggers a signal inside specific cells, like the fibroblasts that form our connective tissue. But IL-11 has a rogue pathway called transsignaling. An enzyme called ADAM10 acts like a pair of molecular scissors. The IL-11R alpha receptor is cut away from the cell surface, becoming soluble. It floats freely in the bloodstream, grabs an IL-11 molecule, and can dock onto entirely different cells that do not even have their own IL-11 receptors. Note that IL-11 belongs to the same family as IL-6, another well-known inflammatory cytokine. Recent studies show the IL-11 receptor can act as a backup docking station for IL-6, meaning these inflammatory pathways can cross over. The presence of both anchored and soluble receptors means IL-11 can initiate signaling in cells that would otherwise be immune to its presence. This biological reach allows chronic inflammation to take hold across nearly any organ system in the body. Now that we know how it docks, step 2 is tracking the damage. To treat the aging process, researchers have to visualize it operating at the molecular level. This image shows a simulated fluorescence microscopy layout of 2 tissue samples. Scientists use a technique with green fluorescent protein to map gene activity. Look at the aged sample on the right. As organisms get older, tissues in the liver and muscle express significantly more II-11, which glows with intense neon green clusters, compared to the dark, young sample on the left. This flowchart shows exactly what that buildup does inside the cell. When II-11 interacts with the node labeled AMPK, a pathway responsible for healthy energy regulation and metabolism, it forces a toggle switch to the off position, dimming the entire pathway. At the exact same time, II-11 interacts with the ERK and mTORC1 pathways. These pathways govern cellular stress and cell growth. Here, the switches are pushed to the on position, throwing the nodes into a state of hyperactivation. Turning AMPK off while leaving mTOR and ERK on accelerates cellular senescence. It forces healthy cells to stop dividing and turn into inflammatory zombie cells that drive systemic, age-related decline. Step 3 is the intervention. To stop this decline, researchers deploy neutralizing antibodies. These are microscopic Y-shaped proteins designed to seek out the IL-11 molecule in the body, physically colliding with it and aggressively capping it so it can never dock with a receptor. The first structural outcome of this blockade happens in the connective tissue. It stops normal fibroblasts from transitioning into myofibroblasts, which are hyperactive cells that secrete massive amounts of collagen. Blocking that specific cellular transition prevents the severe scarring typically seen in the lungs, heart, and kidneys as we age. It intercepts the tissue damage normally driven by TGF-beta, a primary factor in fibrotic disease. This diagram shows the second major outcome. Aggressive cancers, like gastric and colorectal tumors, secrete their own IL-11 to feed their own growth, a process called an autocrine loop. When the antibody is introduced, a barrier symbol breaks the cycle, cutting off the tumor's self-generated growth signal. By deploying this antibody blockade, we sever the central biological supply line that sustains both chronic tissue scarring and self-feeding tumor proliferation. Step 4 is measuring the results of this intervention in a living organism. This dashboard shows the comparison between 2 75-week-old subjects, roughly equivalent to 55 human years. On the left is the untreated control, showing advanced physical deterioration. On the right is the subject that received the anti-IL-11 therapy, displaying preserved health, high muscle mass, and low adiposity. The treated group experienced specific metabolic reversals. They cleared out their accumulations of senescent cells, significantly decreased their total fat mass, and restored their baseline muscle function. This chart shows a Kaplan-Meier survival curve, mapping the lifespan of both groups over time. The line for the treated group extends 25% further along the x-axis before dropping off, representing a quarter increase in overall lifespan compared to the control. These metrics indicate that the intervention addresses specific metabolic drivers of age-related disease. By reducing fat, increasing muscle, and extending survival, the blockade disrupts the typical biological progression of late-life frailty. We have successfully completed the therapeutic blueprint protocol for IL-11 inhibition. By mapping the receptors, tracking the spread of cellular senescence, and deploying neutralizing antibodies to block the signal, researchers took subjects equivalent to 55 human years old and extended their median lifespan by a quarter. This is an active reality in modern medicine. Anti-IL-11 therapies are already entering early-stage human clinical trials right now to treat fibrotic lung disease and thyroid eye disease. This protocol provides a concrete method for intervening in the molecular pathways that dictate how a body breaks down. By targeting the IL-11 signal, it is possible to move beyond treating individual symptoms and address the underlying drivers of mammalian aging.